Thank you, Dr. Smart. You stole my introduction because I've been really impressed with the breadth and width of different fields that we've seen present within high-value nutrition thus far. And I'm going to step a little bit further outside of that and present some work from the fields of biomedical engineering and clinical medicine in respect to high-value nutrition. I was particularly enthused yesterday to hear Dr. Smart mention in his opening address that we need to focus on those consumers that have health and well-being issues, because it's those specific consumers, or as we're referred to them in the health field as patients, that are central to the topic I'm going to be discussing today. The mechanical processes of digestion are initiated and coordinated by an underlying electrical rhythm termed the slow wave. These slow waves propagate down the stomach and intestines in a very routine, organized fashion. You'll see here a video from inside the stomach, where we can see these large ring contractions that we know are responsible and vital for the breakdown and movement of contents of the gastrointestinal tract. Each of those contractions is initiated and coordinated by an underlying electrical slow wave. Unfortunately, most of the previous studies of slow wave propagation have relied on low-resolution techniques, whereby a few electrodes were sparsely placed along the stomach, forcing researchers and clinicians to make assumptions about activity occurring between and around electrode sites and precluding any accurate spatial propagation analysis. We thereby developed high-resolution human electrical mapping, whereby 256 electrodes are simultaneously placed directly on the surface of the stomach intraoperatively to record slow-wave propagation dynamics in very high spatiotemporal resolution. Now, these methods introduce their own set of problems, namely in the vast data sets that were generated. We thereby developed a custom automated software package called the Gastrointestinal Electrical Mapping Suite, which included a number of different automated and semi-automated computational algorithms, which effectively made the translation of this technology to a number of human mapping cohorts feasible. And of those, I'm going to touch on 3 different cohorts today. The first being patients with healthy stomachs. So those are our healthy controls. We then mapped 2 different diseased cohorts, the first being gastroparesis, which is a severe stomach condition where patients suffered debilitating symptoms of pain, bloating, early satiety, nausea, and vomiting. These patients' stomachs also failed to empty properly. The second disease state was chronic unexplained nausea and vomiting, where patients experience the same severe symptoms, but their stomachs empty properly. Now, what we found in these cohorts is that in the healthy case, you can see an example animation here of slow wave propagation. Each square represents an electrode, and illumination represents a slow wave activation at that point. So what we can see here is a very routine, organized slow wave propagation in the healthy patients. This was the case in 20 out of 21 of our control patients, with only one very minor deviation in this very routine activity in a single patient. Conversely, our disease states exhibited a wide range of abnormal or dysrhythmic activation. In this example from a gastroparetic patient, we see a couple of slow waves that appear to be normal, but then we see a very abnormal dysrhythmic activation pattern break out, where we have ectopic events, retrograde propagation, rapid propagation, et cetera, et cetera. It's highly dysrhythmic activity. And the same was the case for our entire chronic nausea and vomiting cohort. In this example, we see an ectopic pacemaker that's located on the incorrect side of the stomach compared to normal activation, and also exhibits additional abnormal propagation characteristics. We're able to interpret these dysrhythmias in a revolutionary level of detail, which also allowed us to develop a new classification scheme for gastric dysrhythmias. Now, from this figure, it's important to note here that a number of dysrhythmias occurred, and actually a large percentage occurred within the normal slow wave frequency range, meaning that they likely would have gone missed or undiagnosed using less sophisticated low-resolution techniques. So at this point, I'm going to slightly change gears to a complementary and parallel research stream that we run, which is our multi-scale mathematical modeling. We're able to develop mathematical models based on a series of complex mathematical equations to represent and interpret our experimental data and expand that data across a number of different scales, including cellular, tissue, organ, and body surface levels. What we're able to do with these models is investigate in silico a number of additional parameters and hypotheses. For example, in this case, we're able to investigate the effects of cellular depletion on slow wave propagation in a tissue level and cellular-based model. We're also able to interpret and expand upon our high-resolution mapping data. In this case, you can see an anatomically specific and accurate slow-wave model of the healthy human stomach. This is based directly on and informed by our high-resolution mapping data. But we're also able to expand that data further, in this case by translating the known biopotentials at the stomach surface to the body surface, allowing us to improve the accuracy of lower resolution or less invasive recording techniques such as body surface potential recording. And finally, the last model I'll show is of an ectopic pacemaker, a dysrhythmia that was specifically modeled off of our work in gastroparesis, but in this case, modeling allowed us to expand that to the effects at the whole organ level and compare it then to the normal propagation on the left. So in conclusion, I hope that I've shown here that high-resolution mapping and multi-scale modeling have provided improved insights into gastrointestinal regulation, function, and health. I also hope that it's shown that engineering principles can be highly valuable in improving our understanding of the processes and mechanisms of digestion. And furthermore, these data and methods may provide an enhanced basis for the research and development of functionally targeted food and nutrition, which leads me to our future directions. We're obviously very interested in the clinical aspects of this research, and we're rapidly investigating and innovating new devices and technologies to translate to the clinical setting, including a number of therapeutical interventions. However, one of the answers for this complex problem of gastric dysrhythmias may actually lie in food and nutrition, where ginger has been associated with improvements in nausea and vomiting, gastrointestinal disorders, and also gastric slow-wave dysrhythmias, the same dysrhythmias that you just observed in a revolutionary level of detail, which I hope brings us full circle to what we've heard Dr. Smart, Cameron Smith, and others reiterate throughout this meeting in terms of the value of health research towards high-value nutrition. Thank you.